Well, good morning, everyone. I want to thank Professor Dr. Wessick and the organizers for inviting me to speak today. I'm very excited to attend this meeting and show you some of the new data coming out of our lab. I have no relevant disclosures. Today, I want to talk about roles for mitochondrial dysfunction in Alzheimer's disease. And to this end, I'll cover the following topics. First, we'll look at how central mitochondria are to various diseases and talk specifically about Alzheimer's disease. We'll also identify some of the mitochondrial functions disturbed in Alzheimer's, and we'll look at three groups of data that are being generated from my lab. First, we'll look at data showing sex-based differences in mitochondrial dysfunction. Then we'll look at some data implicating a regulatory role for the transcription factor NF-kappa B on complex 4 activity. And finally, we'll look at some examples of reversing dysfunction and improving function with compounds such as creatine and nilotinib. Let's talk first about mitochondrial disorders and Alzheimer's disease. So we can divide mitochondrial disease and dysfunction into two main categories, comprising over 30 diseases or conditions. And the first category are the so-called primary mitochondrial disorders, which are more rare and they're genetically inherited. They're diagnosed by identifying mutations on mitochondrial DNA that we see here in this slide, designated by the dark green circles. And we can also group the secondarily acquired mitochondrial dysfunction, which is more common, and these conditions are caused by non-mitochondrial genes or nuclear DNA genes, or also by environmental factors or a combination of the environment and genetics. Distinguishing whether mitochondrial dysfunction is inherited or acquired can sometimes be challenging. And acquired dysfunction includes conditions that we're more familiar with, such as diabetes, cancer, Parkinson's, and Alzheimer's disease. So what is Alzheimer's disease? Well, Alzheimer's disease is the most common form of dementia. Typical symptoms involve impairments in memory, but also other cognitive and behavioral changes are seen. Pathological hallmarks of Alzheimer's include the toxic amyloid plaques and also filamentous tangles associated with tau protein hyperphosphorylation. Aging is actually the greatest risk factor for Alzheimer's disease. However, Alzheimer's is thought to be a multifactorial condition. And in addition to plaques and tangles, there are at least 30 other important mechanisms that play key roles, such as inflammation and mitochondrial dysfunction. Importantly to note, there are sex-based differences in the occurrence of Alzheimer's that we cannot yet fully explain, with about 2/3 of all those with Alzheimer's in the US and Canada being women. Over the years, there have been several hypotheses for what causes Alzheimer's, and in 2004, Swerdlow and Kahn proposed the mitochondrial cascade hypothesis. And in this hypothesis, they emphasized the importance of mitochondrial dysfunction in the pathogenesis of sporadic AD, which argues that amyloid may be triggering changes in mitochondrial function. And over the last 15 years or more, we've observed a number of mitochondrial functions affected in Alzheimer's, and these include increased reactive oxygen species, decreased axonal transport of mitochondria, also changes in mitochondrial shape and size, alterations in mitochondrial quality control mechanisms, also a disruption in mitochondrial dynamics, such as in fusion and fission proteins. It decreases in overall mitochondrial numbers, and also deficits in protein subunits and complexes of the electron transport chain, such as in complex 1 and complex 4. And then finally, also decreases in glucose metabolism. And I highlight some of these points here in yellow, since I'll be showing some of our data that corroborates these findings. So what about brain metabolism? Well, to look at overall patterns of brain metabolism, we've been conducting PET scans routinely in our Alzheimer's mouse models. And in this particular experiment, fluorodeoxyglucose, or FDG, A radiopharmaceutical was injected into both control and Alzheimer's disease mice. And the images here show an overlay of PET scans on MRI scans. And this allows us to visualize not only functional changes in brain metabolism, but it also allows us to see the high resolution brain anatomical structures. And the colored or shaded areas then show areas and regions with changes in FDG uptake. So in this study, we found changes in FDG uptake in the piriform cortex and the insular cortex bilaterally, as you can see in the panel on the right. Using statistical parameter mapping analysis, which involves a two-sample t-test, we found that FDG uptake to be significantly reduced in both the insular and piriform cortex at both hemispheres of the brain. And these regions are associated with olfaction and neurogenesis, which may be affected early in Alzheimer's disease. This data was published last year in the journal Neurobiology of Disease. Also, human studies have found decreases in brain metabolism as patients transition from normal to mild cognitive impairment, or MCI, and then to various stages of dementia, as seen in these PET scans. So, PET scanning is a good place to start in order to identify regions of the brain at risk for bioenergetic changes. In additional studies with our 3xTG mice, we showed changes in several mitochondrial proteins and their expression levels. So, for example, this slide shows age-dependent and sex-dependent changes in dynamin-related protein 1, or DRP1, which is known to regulate processes of fission. Moreover, when we examined mitofusion proteins, regulators of fusion, we again found age-dependent and sex-dependent changes in the expression of these proteins. Overall, when examining DRP1, MFN1, and 2, we found a shift towards more fission. Changes in mitochondrial dynamics can have an important implication for not only for mitochondrial function, but also for memory. And synaptic plasticity in memory, of course, requires energy. This video here, created by a postdoctoral fellow in our unit, Dr. Mohammed Sabir, shows the motility of mitochondria, which is required for mitochondria to move to sites of synapses actively encoding new memories. And disruptions in biodynamic processes can affect memory encoding and development and have been implicated in several diseases, including Alzheimer's disease. We also looked at the uncoupling protein UCP4 and likewise found that its expression levels changed in an age-dependent and sex-dependent manner. And disruptions in uncoupling proteins, which are mitochondrial solute carriers, can affect mitochondrial membrane potential and also ROS levels. Next, we examined oxygen consumption rates, or OCR, in either cell cultures or tissue homogenates and analyzed these samples using the seahorse XF24. Dr. Yelena Dvorževic in my lab, a postdoctoral fellow, led these experiments using both the 3xTG strain and the TG-CRND8 strain, another widely used transgenic model of Alzheimer's. In many of our experiments, we use a standard protocol to modulate mitochondrial function that I'm sure most of you are familiar with. That is the subsequent addition of ADP, ogliomycin, FCCP, and then rotenone and anti-myosin A. In these experiments, isolated cortical and hippocampal mitochondrial samples from both male and female adult mice were tested. Here we found increased basal and coupled respiration in female hippocampal samples, but not in males. Also, decreased uncoupled respiration in both female and male cortical samples. Here I show in another study, this time conducted in 3xTG mice using both males and females and sampling the hippocampus and cortex in 3 different age groups. That is at 2 months, 6 months, and 14 months of age. Here we found decreased mitochondrial function very early in the cortex and hippocampus of female mice, and in fact at 2 months of age, while in males, the OCR deficit appeared later, around 6 months of age. Significance was shown here in these different illustrations by the large red block arrows. Overall, the cortex seemed more susceptible to mitochondrial changes than the hippocampus. And this data was just published in the journal called Cells. We also used the Oxyograph 2K to measure OCR. Using this instrument, we found that OCR was reduced in female hippocampal samples, but not in males. So what causes these mitochondrial deficits? One possibility is that they are caused by amyloid beta, which is thought to enter the mitochondria at Tom sites, or sites with pores called translocase of the outer membrane. Once inside, A beta may interact with specific sites or molecules in the mitochondria. Some studies have shown interactions of A beta with ABAD, also called amyloid beta binding alcohol dehydrogenase, which increases ROS. Other sites include cyclin D and the electron transport chain, to name a few. So with this in mind, we've begun to measure various forms of amyloid in both cytoplasmic and mitochondrial compartments at various ages. So far, we found that there are sex-based differences in the appearance of amyloid oligomers when testing mice that are 6 months age. We're still testing for amyloid in other age groups. And these data here show increases in amyloid in females, but not in males at 6 months. So another area of research in my lab involves NF-kappa B signaling. Importantly, the transcription factor NF-kappa B, a mediator of inflammation, has been found to be co-localized with amyloid plaques in human brain in Alzheimer's patients. NF-kappa B may be another factor involved in triggering mitochondrial dysfunction. For example, we know that Cogswell et al. A few years ago found NF-kappa B subunits in mitochondria. Also, a study by Shi et al. Caught our attention that implicated NF-kappa B in changes of complex 4, the site of oxygen consumption. In the left panel, an MTT viability assay for mitochondrial function was used in HT22 cells. Here, A beta induced mitochondrial dysfunction was characterized by using a range of 5 to 20 micromolar of amyloid beta. When Bay11 7082, an NF-kappa B inhibitor, was also applied, Bay11 was seen to block the A beta induced dysfunction that we can see here in panel A on the left, and also improve ATP levels shown in panel B. In the right panel, A beta applications were also found to reduce COX-3 activity. However, applications of Bay11 reversed the A beta induced reductions in COX-3. As a reminder, this cartoon shows the position of COX-3, which is a mitochondrial DNA encoded subunit. Looking more closely at complex 4, we recall that COX-1, COX-2, and COX-3 are all actually encoded by mitochondrial DNA. And the current dogma, of course, suggests that mitochondrial DNA is primarily inherited along a maternal lineage, which may have important implications for Alzheimer's disease. Studies have also shown that A beta interacts with various electron transport chain sites, including COX-1. So our lab has been looking closely at potential changes in protein subunits and the electron transport chain in general, and in complex 4 in particular. And here in this slide, we show decreases in the expression of NDU FB8 in complex 1 and also in MTCO1 in complex 4. However, when we compare our results in males versus females, we find that only females show deficits in the expression of these subunits. In other experiments using a different strain of mice, the NF-kappa B P50 knockout model was used, and we showed that OCR was altered in a sex-based manner. Also, using the NF-kappa B blocker SN50, we also showed decreases in OCR, further implicating NF-kappa B in complex 4 function. Taken together, these data suggest an interaction between NF-kappa B and complex 4. For further discussion on this topic, you can look at my review paper published in Frontiers in Cell and Developmental Biology. All right, let's look now at some of our work with both creatine and nilotinib. Now, in this study, my postdoctoral fellow, Dr. Wanda Snow, tested mice fed a creatine diet for 8 weeks. And creatine, of course, is an over-the-counter product we can buy at the drugstore, but we also consume creatine when we eat meat and fish. And Dr. Flint Beal at Cornell, of course, has published several papers over the years on the protective effects for creatine and neurodegenerative diseases. So Dr. Snow found that creatine improved learning and memory as tested in our Morse water maze, and also improved OCR as measured in the seahorse and control mice. When using the 3xTG Alzheimer's model, she also found sex-specific effects of creatine supplementation on several parameters of spatial cognition in the 3X model. In addition, she found overall improvements in mitochondrial function that were sex-specific with regard to different parameters of mitochondrial respiration. We also tested the anti-cancer drug nilotinib, and this work was led by my postdoctoral fellow, Dr. Ida Elimbagodam. Nilotinib is a tyrosine kinase inhibitor, and it's also a medication used to treat different types of leukemia. But recently, it's been found to have benefit in Lewy body dementia and Parkinson's dementia in a small clinical trial at Georgetown University. Related Alzheimer's disease work with nilotinib is being conducted by our collaborator, Dr. Scott Turner at Georgetown, and Scott's currently testing this drug in Alzheimer's patients at the memory clinic he directs in Washington, D.C. So to test the effects of nilotinib, we assessed OCR in both neurons and astroglia samples from 3xTG mice. And here we found that 100 nanomolar nilotinib was optimal for improving several mitochondrial respiration parameters as measured in the seahorse. The numbers of mitochondria in 3x TGAD astroglia were also compared in the presence and absence of nilotinib using electron microscopy, and mitochondrial numbers were found to increase at the 100 nanomolar molar treatment of nilotinib. Nilotinib was also found to increase the expression levels for some of the biodynamic proteins such as FMF and 1, but not DRP1. We also found that nilotinib reversed complex 4 subunit expression deficits and also enhanced the expression of subunits in complex 1, 3, and 5 that we don't show in this slide. Lastly, we determined that the effects of nilotinib were NF-kappa B dependent. This was realized by applying the NF-kappa B inhibitor BA11, which showed that its application reversed the enhancement of 100 nanomolar treatment. So in other words, nilotinib-induced OCR increases were reduced in the presence of the NF-kappa B inhibitor BA11. Western blot experiments shown here demonstrated increased levels in several proteins, including CAM kinase 2, PGC1 alpha, and NRF2 in our 3xTG Alzheimer's astroglia with a treatment over 24 hours of 100 nanomolar nilotinib. We found an increase in calmodulin kinase 2 expression in the presence of increased NF-kappa B activity, which is consistent with other literature showing that CAM kinase 2's role in regulating inflammation. These data also suggest that nilotinib may increase the number of mitochondria through a CAM kinase 2 PGC1 alpha NRF2 pathway, which is influenced by NF-kappa B levels and 3xTG astroglia. So taken together, these findings support a role for nilotinib in improving mitochondrial function and suggest that astroglia may be a key therapeutic target in treating Alzheimer's disease. We just published this study in the Journal of Aging and Disease, which can be found online. So to summarize, I hope I've convinced you that sex-based differences exist in mitochondrial dysfunction in our different Alzheimer's disease transgenic models. Also, that mitochondrial dysfunction begins early overall and even earlier in female mice. In fact, when compared to other deficits, OCR deficits appear much earlier before several well-characterized pathological changes in the 3X TG model. Our data also suggests that we contradict the idea that hormonal theory alone increases the risk for Alzheimer's disease in female mice. Also, the complex 4 seems to be more vulnerable to insult in our Alzheimer's model, and that NF-kappa B has influence over complex 4, which might serve as a promising drug target. And then finally, mitochondrial medicines and therapies for Alzheimer's disease need to be sex-based and personalized. So I want to thank not only the 3 postdoctoral fellows that whose work I showed in this talk, but also other members of my lab that contributed in various ways to our research. And then finally, a big thanks to many of my key funding sources. Thank you for listening.